this place. So yeah, I'm going to talk about Pop-Eve, which is actually a project that Johnny Mafalda and I started back when they were postdocs in Debbie's lab a little while ago. And so basically, we build models that learn from evolutionary sequences, because you can kind of think of evolution as like millions of years of experiments captured in much less data than you normally have with a ton of experiments. So we have multiple models that come out of our labs. I'm sure Pascal went over a couple of these already. And we apply them to different areas like protein design, viral escape and vaccine design, and what I tend to do is more like human disease genetics and looking how can we make our models better and more useful for finding candidate variants in genetic diseases. So basically, we want to find like the most likely causal variants, and first we're looking at like developmental disorders. The whole thing from genotype to phenotype, can we actually predict which is the most likely causal variant just from the genotype? So with our earlier models, we basically saw that the scores for genes that were really constrained and the scores for genes that were unconstrained didn't seem to be on the same range that we would expect. You could see that like the most deleterious variants in like the unconstrained genes tend to have scores that are about as deleterious as those in constrained genes. This is because we have like individual models for each protein. So like what's very deleterious for the protein's function may not be deleterious for us like as humans, as whole organisms. So we basically decided to calibrate our model scores using population variation information. So what it does is we use a Gaussian process based off of seeing what missense variants are seen in the population versus ones that are not seen. So this is like does not incorporate any allele frequencies and is like just a very gross picture of constraint. And we can find that after popping our models, we see that Pop-Eve is acting as expected, and the constrained genes tend to have far more deleterious scores than the unconstrained genes, as you'd expect. And what this does is it really gives you a way to compare scores across genes and hopefully be able to rank them by how deleterious you think they are to a person. And also, even though we use population data, it's been found that we have very little ancestry bias, which is pretty great because most of the leading other variant effect predictors do have quite a bit of ancestry bias, especially if they use allele frequencies directly. And this isn't good for anyone that's not well represented already in the big cohorts. Well, we perform pretty well on DMS and ClinVar, like the things in Protein Gym. But especially with ClinVar, there's not much room to improve. So how do we know if we're actually doing better if it's only an incremental improvement over some models and it's neck and neck with others? Another issue is this really relevant to the downstream task of trying to find like the most likely causal variant of a severe disorder, especially since maybe we all have multiple pathogenic variants that could cause a disease in someone somewhere. So what do we actually need to predict with our models in order to find these causal variants? So like underlying Eve and all these evolutionary models, we're trying to find like the protein fitness and like how does that mutant actually affect the protein's function in a good way, bad way, neutral. Then we can also ask like what is the pathogenicity, and often people binarize this into just a label of pathogenic or benign. You probably could break this down into things that are like more complex, like include penetrance and expressivity, like can it actually, like how often does it actually cause disease in everyone that has the variant? So it's not quite binary. And then another thing is like how severe would the phenotype be? So like if a variant in a person, would the disease cause death Or would it just cause like a mild disorder or disease that negatively affects your life, but not reducing your lifespan? So it's like how severe actually is the disease that comes from these variants. So one thing to think about is like what would it actually look like for our models to capture these properties? So for example, this is, sorry, got a bit of cold. This is a distribution of the variant scores, like from EVE or ESM1V or alpha missense. And you can see that it's just like a different, yeah. So what we can do to understand like protein, how it affects protein function is like how likely is it to be seen versus the wild type. So we're basically using this as a way to say that things that are much less likely than wild type are probably impacting the protein function fitness. And things that are much more likely to be seen than wild type are going to be benign, if not beneficial. And so then how would we see about pathogenicity? So with EVE, we basically fit a Gaussian mixture model to give us two distributions, so we can then call something based off the likelihood of being in the pathogenic distribution or the benign distribution, and then everything in between is just uncertain or ambiguous. But then when it comes to severity, could we just use it like based off of the magnitude of the score, like without binarizing? So how would we actually benchmark to see if we can capture these things? So the first two pretty much are covered usually by DMS and ClinVar benchmarks, but there are very few that actually cover things like severity. So how would we do this? And this is what I've been really wrestling with the past year. So one option is to look, do these variants cause premature death? And if so, like how early on in life? When does it onset? Like, is it like a childhood or prenatal disease, or is it something that comes on when you're elderly? Very different levels of severity if you think about like years of life, like quality of life lost. And then like are symptoms more severe and interfere with daily life, but this is a little more nebulous and hard to figure out. So we're gonna focus first on the first two. And so I took ClinVar pathogenic variants and divided them into gene phenotype pairs that are associated with death in childhood and those that are associated with death in adulthood using labels from Orphanet. And we find that with our POP-EVE scores, we see a slight separation between those that cause death in childhood and those that cause death in adulthood. And so if we compare this to other models, we can see that they do not separate these things and sometimes even predict that the variants that are causing death in adulthood to be more deleterious on average than those in childhood. And we're one of the only ones that are able to do this like visual separation. So we wanted to come up with a way to actually do this rather than just like looking at the distribution and guessing if it was good or bad. So we took the, as a beginning step, took the 5th percentile score of the benign variants for each model and basically compute an odds ratio. So comparing the number of variants on the left side of that threshold that cause death in childhood over the number of variants, that cause death in adulthood. And we can see that compared to all other models, our odds ratio is far more greater than all of the other models. And quite a few models actually predict in the wrong direction. And this is all significant. And then we also wanted to look at what about onset. So again, took from Orphanet, things about onset in childhood, onset in adulthood. And here, childhood includes prenatal and natal and antenatal, those things, as well as childhood. So we can see that there is a separation, but it's a little bit less than with the death labels. But with the other models, there's much less separation. You see a bit more with like the primate AI, and even alpha missense has a little bit more. But still, it's just not comparable to the separation that you see here. And that if you were to like build a classifier on this, again, PopEve would do the best at saying that things are childhood onset versus adult onset. So why do we think that these models are doing so poorly while they still correctly predict pathogenicity, so they're doing well on the ClinVar? My guess is that the ones that do a little bit better on these tasks, including our own, tend to be very purely unsupervised. We're just trying to find the variant fitness, while other models are either tailored or they've been fine-tuned for the downstream task of doing these pathogenic benign clinical labels. And it just ends up with this bimodal distribution where things are either pathogenic or benign. Yeah. So we wanted to find a way to kind of get at how do we separate out more severe cases from less severe cases. So one way is to basically compare these children with severe DEB disorders to people who are generally relatively healthy in UK Biobank. They'd probably have if anything common diseases. Some have some genetic disorders, but they're all living past their 40s. It's a study of aging, so they're doing pretty okay. So what we did is we took the most deleterious variant per person, and we expect that with increasingly deleterious thresholds, our scores should recall more of the cases versus the healthy population. And so we first looked at looking at de novos from the cases and whole exome sequencing from the controls. And we see that we recall quite a bit of the cases before, and also these cases are subset to ones that we think are caused by a de novo missense variant using mutation rate and expectation of the background. We see that we recall quite a bit more of the cases versus the general population, and compared to other models, we see that they think that a lot more of the general population has variants that are as deleterious as variants that could be causal of a severe genetic disorder. And one of the reasons this might happen is that if you're going towards this bimodal distribution of scores, trying to get things to separate out pathogenic or benign, you get quite a bit more in the pathogenic area, even for the general population. So if we were to take alpha missense's recommended cutoff for pathogenicity, you'd actually see about 90 pathogenic variants per person in UK Biobank. So it's just as a result of trying to go for this pathogenic or benign, we're kind of losing our generalizability to the general population and beyond just disease genes. And so when we actually look at whole exome sequencing for these cases and compare it to the whole exome sequencing for the UK Biobank, we see that we have a more one-to-one relationship, because we expect a lot of the inherited variants in these cases to not be that bad. Often, they're not causing their disorder. I haven't subset to ones that we think a missense is causing the disorder because we can't really do that same estimation. But we can see that in the bottom left corner, we actually get 100% of the missense diagnosed cases far sooner than any other model. So for about 100% of those cases, we get less than 20% of the UK Biobank as having something as deleterious, which could be better, but it is far better than any other model. So then we asked, could we do this on an individual level, comparing the de novo variants from a case and their inherited variants. So if a patient were to walk into a clinic and you only had their sequencing, could you find the most likely causal variant, even if you didn't have the labels? So we took the most deleterious de novo variant and the most deleterious inherited variant. And if you were able to properly rank things by severity, and you think the missense de novo is causing the disorder, that should be much more deleterious than the inherited variants in that person. So we can see that, so at this point, this is our severe cutoff for PopEve. We find that if we use that cutoff, we get 98% of the people that, or their de novo variants back as the most deleterious variant versus all of their other inherited variants. So even without that label, we would have been able to find what we think is the likely causal variant. And you can see that with other models, they quickly drop off and aren't able to rank the de novo variants as the most deleterious. As we increase the threshold, they get even more and more inherited variants that are ranked as deleterious or worse than the de novos. So this is like hinting that we can even do things without trios, maybe try to find the most likely causal variants if we had like a very strict threshold. And this could be very useful for people that don't have trios completed to identify possible causal variants. So basically, I wanted to come around and say, like, we think that PopEve versus other models is better at capturing this notion of severity, even if that's not the proper word to use in this context. And if you think there's a better one, please tell me. And so we're able to, separate out these like childhood death and onset better than any other model. We don't overpredict deleterious variants in the general healthy population compared to other models, but there's always room for improvement. We find their variants in general more deleterious than those in healthy children, de novo variants. And we also find the de novo, likely causal de novo variant as more deleterious than the rest of their inherited variants in individual cases. So just a little overview of PopEve that I didn't really cover any of our other benchmarking or our possible discoveries. But we perform well on ClinVar and DMS. We're state of art of distinguishing severe cases from controls, and this is shown in some other independent studies as well. Minimal population bias. Hopefully, I've convinced you state of art at variant severity. And we find evidence of over 100 novel candidate developmental disorder genes. And you could read more in the paper about this. And also, since pre-printing this about 2 years ago, about 20 of these genes have since been added to Decipher, so they've been discovered simultaneously while I've been waiting to get published in other venues and other cohorts probably. So in sum, we want to be able to build models that can capture multiple properties of like variant effect and function and not reduce our scores to like these binary clinical labels that may not be very useful in the end. And ideally, we could even have like a multi-dimensional score. Like, could we capture inheritance patterns? Could we capture like penetrance and expressivity? Is there a way to like have this already in the data? We just need to find a way to disambiguate and disentangle all of these things and maybe make things useful and a little more interesting. So thank you to my lab and Johnny and Mafalda, who have hosted me and this conference. Helped. Yeah, thanks.